Vancouver. He did her postdoc with Matthias Lichtenfeld at the Dragon Institute, and she's actually the person that developed MIPSI, right, which is one of the really high-level fancy sequencing assay that was used for a lot of high impact publication. And she has used all of this sequencing technology to characterize biological samples. And well, we're excited to have her here and talk about HIV reservoir genetics.
I'm excited to be here too. All right, thank you so much, Sarah, for a very kind introduction. Thank you for having me here. My name is Gwen, and I basically my entire career is in HIV research. I did my PhD in Vancouver, British Columbia with Richard Kerrigan, and I have a very big focus in drug resistance when I was a PhD student. But at the time, I also picked up a lot of deep sequencing techniques. Deep sequencing just started rolling out at the time. So deep sequencing, and then so I got into the wet lab, the chemistry of deep sequencing, and also a lot of molecular epidemiology related to drug resistance, transmission. And then at the time, I had a very strange observation because I was trying to sequence HIV genomes from plasma. You're good, keep going. So I was trying to look at plasma drug resistance versus DNA drug resistance from the same cells, and they don't match up. And that was so long ago, and I was like, wait, the plasma resistance has to come from somewhere, and I didn't understand, and so that actually triggered my entire interest in HIV reservoir research. And so I moved to, after I graduated from my PhD degree, I moved to the Reagan Institute, where I applied all of my bioinformatics, molecular assays development, and also epidemiology sort of skills at the Reagan Institute. I did my postdoc there, and then I was very fortunate to be recruited to Cornell in 2019, and that's where I've been since then. So my research focuses on three main things: HIV reservoir genetic diversity, which is what I'll talk about today, but I also spend a lot of time developing assays, and what I believe is you have a scientific question, and it's very important to actually have the right assay to answer the specific scientific questions that we have. And so having an assay that is custom fit for your scientific question, I think, is one of the things that I'm aiming for. And of course, the global health impact is one of the main things that I focus on. Global health in terms of looking at different HIV subtypes all over the world, making sure that the assays are broadly applicable to different subtypes, because the epidemic is not just in subtype B dominated regions, it's also everywhere else related to different subtypes, but also the clinical impact of the research. Sorry for this very slide. So today I'm going to talk about the DNA story and RNA story and how we can, as an example, I'll bring up an example. How do we bring it to the clinic? DNA and RNA stories in the virus. Okay, so the HIV life cycle. You're probably all very, very familiar with this. But I'm going to focus on the very specific aspects that my lab works on. And okay, so HIV is a fresh virus. There's an RNA stage and a DNA stage of it in its life cycle. So for the DNA, it's an integrated, usually occurs as an integrated form. So you have cell-associated viral DNA, and this gets transcribed into different viral transcript isoforms, and they become cell-associated viral RNA. And then these viruses, if the viral genome it translating all of these viral proteins, it gets packaged into viral virions, and it buds on the cell and circulates in the bloodstream, becomes plasma viral RNA. And this is the marker for viral load for when people are bioanemic. That's what we measure. And so studying all of these three types of DNA and RNA and plasma RNA is very different in terms of the individual that we have to, the technology that we need to use. And so I will make a very clear distinction about these three components. And of course, we have a lot of different ARVs that target different stages of the viral life cycle. None of these can reverse the HIV integration step, so none of our treatment can eliminate HIV from our body. Now, of course, this is complicated by the fact that the virus mutates a lot. the virus has a very high mutation rate. In fact, I quote Doug for this. He got his Doug test book when I was a PhD student. So that's why I've been a big fan. But the viral mutation rate is extremely high, and for the RT reverse transcriptase enzyme in HIV, it makes one mistake per HIV genome. So the HIV genome is very small. It's only 10 kb in length, and the reverse transcriptase has an error rate of 1 error per genome. And so when you activate the virus actively replicates in a person who's viremic, who's under antiretroviral regimen. And so you can have this huge pool of mutants within a single person's body. And this is the perfect Darwinian pool of selection. You have any selection pressure, be it a drug selection pressure, be it an immune selection pressure, autologous antibodies, CTL, or for example, BMAX interventions, all of these would just quickly select for a viral strain that is resistant to it. Now, of course, this high level of viral diversity also translated to having different viral strains. HIV is very different from COVID and very much linked to how HIV is transmitted. In COVID, we are all experiencing COVID. We have the Omicron strain suddenly swept over the entire globe. But for HIV, these mutations translate into different strains that are geographically confined. Subtype B is mostly circulating in North America, South America, Europe, and Australia. But in other regions in the globe, we have all of these other HIV subtypes circulating. So they are they are genetically very different. Subtype B only accounts for 10% of the global epidemic, whereas non-B subtypes account for 90%. Subtype C is the highest, the most dominant subtype. So when I say they're still genetically very different in all these subtypes, so do they actually matter biologically? And so in this slide, I'm trying to convince you that yes, they probably matter. So if you compare subtype A and D HIV in Uganda, these are co-circulating HIV subtypes that are in the same community. People are receiving the same healthcare, the same infrastructure. They have the same host genetic background. And so we can compare them head-to-head and look at people who are infected by subtype D HIV. They have lower pre-therapy CD4 count. Subtype D infections are also associated with a higher mortality rate, faster disease progression. And so in summary, subtype D is more aggressive. So biologically, yes, there is a difference between subtypes. So now genetically, how different is different? This is a phylogenetic tree, and you can see that, for example, here, subtype B and D they are the closest neighbors. If you only look at subtype B alone, the genetic diversity in subtype B reached between 8 to 17% of donors, and this is gap-hole data. So, and this estimate is on the higher end, but it shows you how much just within a subtype, the genetic diversity can exist. And if you look across subtypes between subtype B and D, the genetic diversity goes up to 17 to 35% in the amino acid level. Yeah, this is on the higher end of this spectrum, this particular estimate. But there are cases when a third of the protein was different between two HIV strains. And so it's a very mind-boggling thing for me when I look at these numbers. And then I also look at sequence data, how HIV virus still maintains its own identity as HIV, how does it still function as a virus? And so my research interest is on studying how this HIV diversity impacts clinical outcomes, treatment, and reservoir.
Can I ask you a quick question?
Of course.
That you're talking about DNA or RNA sequence diversity, not amino acid diversity, when you quote 17 to 35%.
This is amino acid level.
That's amino acid level.
Amino
Okay,
acid level.
all right.
And so and that's why I think these estimates are really on the high end, but still it's and looking at, because I've seen a lot of sequence data myself, and it depends on the region that they are looking at, it is still. So, viral genetic diversity. Why does it matter? Because it leads to differences in drug-resistant pathways. I talked about antibody resistance, immune escape. Viral replication fitness, perhaps one viral strain replicates better than other viral strains. But of course, my research these few years has been focusing on the reservoir landscape and viral transmission control. So this is the DNA part of the story. Let's talk about HIV DNA, the covirus. So this on your right here, you see this is what we call the biograph. It's not showing up as an intended figure, but the general gist of it is the dark line, each line on this virogram is a single molecule of HIV viral genome. So we sequence almost end-to-end the viral genomes. So this assay is called FLIPSeq. So this is the first assay that I developed at the Reagan Institute. It's a single copy assay. So we do limiting dilutions according to Poisson distribution, and we sequence one HIV viral molecule at a time in each well. And so for this one study participants, we captured around 63 genomes here. Very expensive assay, it's very labor intensive. So getting to the 63 viral genomes, you can see that almost no genomes are full length. And I only colored the one that is full length and without any premature stop codons, without any hypermutations. I colored that in dark. And so you can see how rare full genome intact, potentially replication competent viruses are in individuals who are living with HIV on long-term suppressive regimen. So there's of course well-defined genetics pipeline to determine which viral genome is intact versus defective. So in this presentation, you will hear me talking a lot about intact genomes, defective genomes. The intact ones are the long ones without any mutations that is associated with effectiveness. And of course, we also validate this using Q-level sequences, knowing actually that this is replication confidence. And so we infer it, we go back and forth between bioinformatics and wet lab data. All right, so that's subtype B. That's my work at the Reagan Institute. And after that, when I started my position at Cornell, and because of my focus as a PhD student too, I work a lot with global HIV subtypes. So one of my main interests is really to transfer this technology and to look at HIV reservoirs in different subtypes. So the very first question that I asked was, so in subtype B, we know that there are very, very few intact viruses circulating in a person's PBMC. But what about subtype A and B? Do they also have very few intact viruses in terms of proportion relative to defectives? And so we looked at the Ugandan cohort. So the study was published last year in Nature Com. And what we saw is on the left, you have subtype A1, and on the right, you have subtype D. Yes, very much like subtype B, subtype A1 also has very few intact defective virus ratio, and subtype D also has a lower intact to defective virus ratio. We tried to do statistics to compare whether there's a subtype difference in terms of intact versus defective ratio between subtypes, and it wasn't powered enough. Just getting the sequencing depth. I'm showing you two of the individuals that we have deep data, and it's just, but you have to go through a lot of wells to get to this level of depth. So, but we did see something interesting. For example, we saw that subtype D is more clonal than A1. So by clones here, what I mean is, so back in 2017, one of the studies, we showed that HIV infected cells can clonally divide. So an infected cell can just multiply and do its own cell thing, but it carries the reservoir with it and it basically makes more copies of the reservoir. So that's clonal extension of infected cells. Now, all clones would have identical sequence. And so we looked at identical sequence and we used that as a proxy for clonal expansion. We later verified this proxy using integration sites. And so there are calculations on how mathematically variability that sharing two integration site is not from the same clone. So usually sharing two integration site of the viruses is a gold standard of defining clonal expansion. And here we use sequence identity. So we saw that subtype D has more identical sequencers, identical viral genomes, meaning that there are probably more clonal expansion happening in subtype D. So this is one of the findings, but of course we are limited by our sampling depth in subtype A1. We only have 94 total sequences from 4 donors. And so there are limiting factors, and so this is not a final biological message just yet. We really want to get more data on subtype D versus A1 in order to see whether subtype D is really more clonally expanded than A1. But regardless, going back to the study hypothesis, we did see that both subtype D and A are associated with proviruses, both defective and intact, that can clonally expand. And so clonal expansion is one of the mechanisms that drive persistence in subtype A1 and D, A1 and D, as it is with subtype B viruses.
Sorry, that's
Of
my
course.
question. Do you have any information on the phenotype, self-phenotype of the clonally expanded cell? That differs among the.
Right, so that's going back to the first study where we did, so not on subtype A1 and D, but in this particular study, we sorted the cells into Th1, Th2, Th9, Th17. So and another technical aspect in this study is at the time fixing the cell and performing these kind of long range sequencing has been super difficult. And so this is one of the technical failures that we broke at the time, is because we had to fix the cells. So basically I did a time series. You fix the cells for a different amount of time and you try to sequence them arbitrarily, which condition gives you the most sequence basically. And yes, so we found that Th1 cells is associated with more clonal expansion. But it makes a lot of sense. Th1 for infections. I think although I did not know the antigen specificity of the Th1 cells. But we didn't have enough cells, and these are really rare examples collected from loci, so we didn't have enough cells to get to those questions. So this, actually, I keep going back to this slide. So this slide, we actually went through 1 billion cells in order to get the Th1, Th2, the entire portfolio per person. So it requires a lot of cells, and the infrastructure is just not there in a lot of African communities. So, and then the African communities and the subtype circulating, there's also extremely complex. So initially, our collaborators have provided me with these subtypes data. They have general RT viral subtyping information, and then we do full genome sequencing, and we found that actually a lot of them are recombinant, but a lot of them are recombinant. So if you look at one part of the genome, this is across the HIV genome, if you only look at RT, and especially in the recombinant part, the RT can be blue, which is subtype A, but the individual can be B. And so working with HIV and recombinant is a very interesting challenge that I see in these studies. For example, if you're thinking about developing an assay specifically for A, and a person walks into the clinic, you don't know whether the person is actually going to be A or D or recombinant. So that primer might not work for across subtype. So we really need something that is cross subtype, but the challenge in getting there because of all of these sequence variations. And I think if anyone of you here is familiar with the IPDA assay, you probably have heard of how difficult it is to get the IPDA assay working across subtypes. So the IPDA assay, so of course, it failed. So the original IPDA was designed based on subtype B sequence. I should explain. IPDA assay is a ddPCR assay that quantifies the copy numbers of intact viruses versus defective virus. So it's a ddPCR proxy. It's an estimate of what is intact and what is not. But of course, it relies on parent syndromes. And for the work high cohort in Uganda, at least I'm showing you 2 failed cases of the general IPDA. And so in this Nature Communication publication last year, we modified the assay by incorporating degenerated basis in the parent syndromes, and we were able to rescue the original assay. we applied is all of these sequencing technology, including FlipSeq, the full genome, SG, viral sequencing, and also IPDA into subtype C cohorts as well. And so subtype C, this is, so I'm very honored to be able to work with the French cohort in South Africa. And we did a lot of viral genome sequencing first. So the study is led by Kavita Reddy. Dr. Reddy is works with FOMB, and so she basically came to Reagan and we worked on all of these viral sequencing protocol together. And then I did the bioinformatics work, and you can see that it's very similar to subtype B, which is the middle pie there. Chronically treated subtype C infections also have a very small fraction of proviruses that are genotypic N-type. And compared to acutely treated and untreated infections, you see a lot more viruses that are intact. In color scheme, you're looking at the dark navy blue. Okay. So this sequence of all of these from subtype C, we also saw clonal expansion, and we also use the sequence data from subtype C to develop an IPDA for specifically for subtype B and C. All right, so all of these. So using FlipSeq, and you generate all of these data, and it's just so far I've just been talking about viral intact and viral intact genomes and defective genomes, but there's so much information on the single viral genome that you can explore. And so the next question that I want to or become interested in is what about the viral elements that controlled by the transcription, for example, viral promoters, and TAR. And so I have very many studies, a series of 3 studies that look at each component here. The viral promoter is located in the 5' LPR. Viral TET binds to viral TAR, which is RNA secondary structure in the beginning of the mRNA transcripts, and it promotes viral transcript elongation. Right, so asking, are the subtypes different when you look at the transcription regulator elements in the proviral genome itself? Is there a possibility that some viruses have a more active viral transcription compared to the other? So this is the promoter, and here I'm only counting NF-kappa B binding sites. And it's very well known in the literature that subtype C has more NF-kappa B binding sites in the HIV promoter. And NF-kappa B is, of course, a human transcription factor. It binds to the HIV promoter, it promotes HIV transcription. And subtype C has more. Subtype B is known to only have 2 NF-kappa B binding sites. Subtype C has 3. And subtype AE is known to only have 1. Now, of course, we also look at the sequence diversity and we ask, well, HIV mutates a lot. And all of these mutations might generate new sites, might result in loss of sites. And what if there's a single site that is mutated relative to the canonical NF-kappa B motif? Is it still going to bind? And so we went into the literature and we found data where people actually mutate like single sites in canonical NF-kappa B motifs, and they have affinity data to show whether a certain site binds. And it's not black and white, if there's a range. And so we have affinity data, and anything that has been previously shown to a motif that binds to NF-kappa B, we count them. And so in these 3 subtype B donors, you can see that these are just the intact genomes. Within each donor, for example, donor 14, there are 2 different NF-kappa B genotypes. Like there's a promoter with 7 sites, and there's a promoter with 8 sites. And so there's intra-host diversity, and there's also inter-host diversity. Now, if you look at the affinity, you can also see that there are differences in the binding method. All right, so I'm jumping to the next. So basically, bottom line is, yes, subtypes are different in the promoters. Are the subtypes TAP different? Yes, the subtype TAPs are also different. For example, in subtype D, HIV TAP is mostly 86 amino acid in length. So subtype C has shorter TAP. A, B, and C usually have TAPs that are 101 amino acid in length. And of course, we also saw differences when we go really into the sequence analysis. We saw differences in terms of the actual mutations themselves as well. That defines the subtypes. All of these are from genome phenotype viruses. So yes, TAP is different. What about TAR? And TAR is also, this is like a mini pet project that I started. And so TAR is a secondary structure in the beginning of the HIV transcript. That's where TAP binds. And TAR is also structurally very different. And so here we use a RNA secondary structure predictor called crypto to predict the TAR structure based on the viral sequences. And so here we're just showing you reference sequences here, but I want to make the point that the two nucleotide or three nucleotide bunch where I highlighted is where TAP binds. And so you can see that the different subtypes already have different structures. And some of them has this 2 nucleotide bunch and 3 nucleotide bunch. And so all in all, we get to hypothesize, and it gives us rationale to hypothesize that the different subtypes are controlling HIV transcription differently, or they will have different levels of HIV transcription. Right. So part one conclusion. Like subtype B, subtype A, subtype C, and different viral genomes are largely defective in chronically treated virologically suppressed individuals. Their extent of clonal expression may differ between subtypes. We are still collecting more data there. Viral polymorphisms might impact IPD success. So again, this talk is about viral genetic diversity. And so the viral genetic diversity, beside clinical impact, beside biological impact, assay impact is another thing that is big important thing to be stressed in a lot. And then the subtype has distinct promoter, TAP and TAR, which may impact reactivation. So some very straightforward questions we can ask is, with all of these genetic diversity, would subtype C react to LRA stimulation, latent reversal agent stimulation, in the same way that subtype B would about an AMD or about recombinant virus? So there are a lot of questions that we can ask, but the bottom line is I want to show you that virus subtype probably matters in your research in many different ways. So sure, we looked at DNA and we think about viral transcription. So viruses actually transcribe at different levels. So that's the immediate questions that we want to ask. Do they respond differently to different LRA stimulation? So this is the RNA story that I want to tell you today. And where's the one time? So the RNA stories that I want to tell you today focus on the cell-associated RNA. So plasma RNA, people have been sequencing RNA, plasma RNA for years. But cell-associated RNA and sequencing of cell-associated RNA is a relatively new arena. Not a lot of people are able to capture full cell-associated RNA, especially from clinical samples, because it's rare. Not a lot of clinical samples have full RNA present at all. And so I'm going to talk about many tools. So going back to one of the things that I mentioned before is, when we have a scientific question, I always try to make sure that we have the most good tool to answer the scientific question. And so here, I'm going to use tool development. So RT-ddPCR. So using a droplet ddPCR method, and on the left there, you can see that there are different types of viral transcript isoforms. Different viral transcript isoforms are responsible for different viral proteins. For example, the unspliced form is the gap hole transcript, but it's also the transcript that gets packaged into the cell. It's a long story, the distinction between what goes into what. But there is the VIP transcript, the VPR. So all of these relies on splicing. So the virus hijacks the whole splicing mechanism to get all of these viral transcripts. Now, there are several studies that shows the sequencing of these entire transcriptome, the viral transcriptome, mostly in cell lines, because it's just very difficult to get the full paper from clinical samples. And so that's exactly what we're trying to tackle here. But first, we do ddPCR quantification of how many transcripts there are. And one of the distinctive feature of this particular ddPCR protocol is the use of oligo-DT primers. So a lot of protocols and a lot of clinical trials that are measuring cell-associated HIV RNA use random hexamers. And so for random hexamers, you'd be also quantifying aborted transcripts. So even if a transcript just goes on a little bit, we still quantify that, which contains the two, or whatever regions that you're targeting with the assay. But so here, we used oligo-DT for total mRNA extraction, and then we use oligo-DT for cDNA generation. And then we use ddPCR to quantify the content of gap hole, the transcripts containing gap hole, which is a count of unspliced transcripts. And then we use another primers and probes to quantify the end of the transcript, which is a count, which is a proxy for total transcripts. And here, on the right here, you see that the green bar is ex vivo samples, so no stimulation, we just took it directly from a donor. So it's not shown as I wish it to be, but the first three donors are viremic. The second donor is a low level viremic donor. Donor 3 to 7 are biologically suppressed. And here you can see that of the 5 virologically suppressed donors, two of these donors have detectable green bars. It means that at least for the number of cells that we have sampled, virus is not latent. So latency is probably a term that we might want to rethink about, because latency implies that the virus is not transcriptionally active. So these people do not have any viruses circulating in the blood, meaning that we think that the virus is latent, but it's really not. It's a cell-associated RNA component.
So when the graph here is looking at the DT primer set, not the GAG-Paul primer set.
This is, oh, that's a good question. I only show, this is the 3' end actually.
Okay, so these could be early transcripts.
These can total transcripts.
Yeah,
Total, but polyadenylated transcripts.
sure, but like, they could be multiple spliced.
Yes,
Yeah.
absolutely. So it can be anything. But then I will, the next slide is going to be on sequencing. So I think, yes, sequencing probably will give you the final answer to this. Also, one thing about using oligo-DT is mispriming. So we had a lot of mispriming issues as well. And so sometimes it's difficult to discern. HIV is a very high AT content, meaning that it's just very difficult to discern which one is a mispriming given. All right, so addressing the question. So which one of these are full length, which one of them are multiply spliced? So in this particular study, we took 115,000 cells from a person who is viremic and who is living with subtype B HIV, pretty high copy numbers of viremia. And we look at cell-associated RNA, and we put this through PacBio sequencing. And this is probably one of the higher resolution HIV cross-sectional HIV transcript profile that you would find out there. So in the first, we saw 3 copies of genome intact transcripts. Genome intact transcript means that these 3 transcripts probably originated from genome intact providers. So with the full sequence, we were able to use the same bioinformatics pipeline to decide whether it's genome intact or defective. Now we also see very clear splice patterns, canonical HIV D1 site splice into A1, D1 splice into A2. We also saw the multiply spliced transcripts there. But this is from a viremic individual, and you can see a multiply spliced transcripts. But the interesting thing is these other colors, the blue. So this is a D1 to A7 spliced, but if I look at the viral genome there, so there's a map there. It's hypermutated. And so, and a pullback imaging could have impacted, could have created a lot of genetic mutations in this genome. And it's hypermutated, but it's still splicing. And it's still producing growth transcripts in this one person released. And not only that, it probably did something to the poly-A signal, and it ran through and it went all the way into the human genome, and I was able to see the human gene and its polyadenylated and the human genome. So HIV is doing something very strange. That's the hypermutated spliced transcript and run through or run again. And then we got the yellow. The yellow is an HIV host chimera. So HIV D1 splice donor site is a very strong splice donor site. And this, in this example, it's spliced into the host gene. So the host gene here is WDR47. And it's spliced into exon 11 for some reason, into WDR47. And so that's another thing that we are. So I have a student now who is specializing in looking at all of these viral host chimeras so that we can understand more about how or how frequent this happens and what type are they of. This is a very interesting observation to me. And also, of course, we were able to see integration sites. So these were not hypermutated genomes, but then it ran into the host genes. So we actually get a polyadenylation in the host at the host end, but then we have hard virus in. This is basically showing you a method using PacBio to look at viral transcription. Yeah, PacBio is really expensive. And in order to get this is only 42 genomes on this slide. We did 4 PacBio runs and many, many thousands of dollars. So it's always a concern with undergrad, so I have to write. I think you are all very with this concern too. So in fact, it all came when I have some samples, clinical samples that I want to analyze. And there's almost no detectable HIV cell-associated RNA in there, but there's some. There's like not none. But if I put it through PacBio, I would just have to sequence so deep and I almost forget about my wallet. So I started developing a, sorry about this. So I started developing an SGA method that it's basically a very similar limiting dilution method that relative similar to FlipSeq. And so instead of DNA, we're just limiting diluting RNA. But it has been extremely difficult so far to get full-length amplification of HIV RNA. Not a lot of studies have shown a single molecule full-length amplification of HIV RNA. And that's for the last few months. Actually, I just got that gel last week. And we actually got 9 kb amplifications, which is almost, these are all unspliced transcripts. And so this is one of the 10 covert here. But if you sequence them with something like this, you still get a very PacBio-like picture where you get unspliced and spliced transcripts. Okay, so and then we move this into single cell. This is a single cell sorting of a cell line called 8E5 lab. And the 8E5 lab cell line is infected with a single copy of HIV provirus. And this HIV provirus has a very known, well-established integration site. And when you culture 85 cells, these culture supernatant is P24. There's so much P24 that you need a huge amount of dilution for analyzing. So meaning that I thought all of these 85 cells are going to be actively expressing HIV. But no. Of course, we didn't find that. So on the, actually only focus on the picture on the graph on the leftmost, we sorted 48 cells into 48 different wells, and we do mRNA extraction from each cell. And so we're dealing with a single cell with single cell mRNA. And the first thing I want to ask is, can we even do that? And yes, we can. So each of these dots is a single cell, and with the extraction, it's very tiny, with 10 microliters, but we split it into 3 different reactions: the 3' LTR transcript quantification, the 5' transcript quantification, and a human gene. So there's a housekeeping gene. Every single well was targeted for a housekeeping gene, meaning that the extraction was successful, first of all, but the HIV transcription is not there, the same. The extraction is successful. Only 22% of these cells are positive for HIV RNA, meaning that even though the culture supernatant is filled with P24, the virus is actively producing GAG protein, but only a portion of these viruses are. And they are all genetically identical. Same integration site. Yes, sir.
So I could be mistaken, but I don't remember, 85 cell one, but initially had a single integration. But as this cell line is propagated and it's infected with a replication competent virus, it starts accumulating integration sites and other genes as well. Everybody's cultures are so that you may have some new integration sites that are not in the active data.
But I've also seen that I actively, I actually tried submitting that to Cory and they didn't like it. So yes, I've been working with the 85 cell line for a very long time, and yes, after a prolonged culture, and because I also do integration site sequencing, we have seen an accumulation of new integration sites. So, and in this particular experiment, I asked Nathan, this is actually an Nathan actually, he is Brad's postdoc and he's working on this project. Brad and I co-mentored him on this. I told him to use the first ball, not even the first ball, like the fresh NIH vial. So without any passage, zero passage. And so we have sequenced the HIV integration site from that particular vial, and it's, I have not seen a non-canonical integration site in at least this particular vial. And so the assumption is they have the same integrations. And so yes, we control for passage here. But the one limitation, I think, is that this assay is an RNA only assay, so we're not able to look at the integration site. We're not able to look at the whole genome of this particular assay. So what I'm going to do with this, all of the technologies of all of these assays. Marina, Lily, and myself got together one day, and then we decided that we should probably, this is our meeting notes. So I was just scribbling on screen. So this is what happens when three scientists come together and then try to figure something out. So with all of these technology, we can already immediately start to look at interesting questions. One of the things that I really want to look at is virologic rebound. So this is, at least I'm trying to draw a person viral load trend after ATI. So before ATI, so antiretroviral treatment interruption, the viral load is suppressed. And then after ATI, after a certain period of time, it rebounds and then you have phosphoviremia. Most studies have used pre-ATI samples to look at association between, for example, total DNA reservoir load and DNA reservoir load with the time to viral rebound, and of course, a lot of biomarkers do. But these people are increasingly studying the time leading up to viral rebound during ATI. And that time period can be very short. So it depends on which study that you are talking about. The sample can even not exist. And so this becomes a very interesting question. But we are very lucky to work with Maria, and she was able to provide us with very interesting samples. So my hypothesis here is, and sorry about the very messy scribble here, that immediately before rebound, I would see a higher activity of unspliced genome intact viral transcripts that are circulating in PBC. That we'll see an increased viral transcription activity longitudinally. So that's the first hypothesis that we want to test. And then also, of course, we are sequencing the plasma to see whether the rebounding virus has any allogenetic relationship with the ones that are in the DNA compartment that becomes transcriptional. These are the three hypotheses that I just talked about. So basically, can we use the presence of genome intact HIV transcripts in PBMC to predict that rebound is in there? And Marina is was very kind to provide us with these HBR trial samples. And so the 4 samples are from COVID 2018 studies, and one participant is from the Mendoza 2018 Nature study. They have all received BNAP intervention, one before ATI and one during ATI. Everyone revamped. Actually, we only have one single participant where the time needing to rebound is long enough that we can actually draw meaningful conclusions to it. But after we did it for all, look at the time to rebound is really, really short. So we're dealing with 4 weeks, 6 weeks, it's so small. This is 4 weeks to rebound, 3 weeks, 4 weeks, and that's 15 weeks between ATI and rebound. But this person did rebound from the authentic code. Very, very much, probably due to the rebound management. Okay. So in that donor, so I thought we are going to see a general uptrend. This is not showing as I intended. This is actually, it was a colored figure. So there's the 3' and the 5'. But the paired bars, the first bar is the 5' and the second bar is the 3' cell-associated fusion start modification. So I thought I would see a trend up leading up to rebound. And then the rebound is at the last time point here for this, the top 3 graphs. But of course, everyone seems to be doing it. Donor 601 is probably the only one that we see an upward trend. In donor 9201, it's almost a decrease. And so, unfortunately, this hypothesis may or may not steady.
Do you think some of this is limited by the fact that you're just sampling PBMCs and maybe?
So these are all PBMC samples. So yes, this is one of the biggest limit.
But Brandon Keel has shown that the reactivation occurs primarily in the gut and ultimately the tissue.
And then this presentation of Cody about one cell. That was amazing. And so using blood as a predictor of viral or rebound in plasma may be challenging it. We are almost here. I'm almost hoping for that there's a rebound in a certain compartment and spreads, and it becomes more apparent. Okay, so HIV transcript sequencing. So, but with all of these precious samples, before and after ATI, what we're trying to do is look at the viral transcripts in them. And so, good old gel, SGA gel, so the limiting dilution. So we were trying to capture viral transcripts. So same sequencing approach that I've shown you before. And very interestingly, all of these viral transcripts are super small. We are not able to see anything that is longer in these individuals. And so we're still in the process of sequencing these particular transcripts, so I will have more updates later. But with all of these around public, I've got the results of really particular very exciting findings. And there's also, looking at time, about 10 minutes, I think we should be on time. So finally, the rebounding plasma viruses. So remember, all of these individuals experience virologic rebound, and we're trying to look at the viral sequences that are circulating in the plasma after rebound. And going in, I thought the viral sequences would be very homogeneous. They would probably all come from the same source, but of course not. HIV always surprises us. There are so many quasi-species within the plasma compartment. And again, this technology, or this particular sequencing run was done before we have the SGA4 genome runs, and so they were not full genome. They were sequenced in 3 different chunks. But for each of the sub-region of the subgenomic region of the virus, you can see that there are a lot of quasi-species circulating. Okay, so part 2 conclusion, and I have one slide for part 3. Viral reservoir is not truly latent, and I think a lot of cell-associated RNA studies have shown us that we can detect viral transcription activity in a lot of individuals who are on suppressive therapy. The transcripts tends to be short, or at least so far, the transcripts that I saw were really, really short. Meaning that they are probably either multiply spliced or it's abortive or something strange, or maybe it's a chimera. of the host. The patterns are different across donors in subtype B, and the rebound viruses are very diverse. So here in this particular part, I want to show you that we have now the tool to full genome, full grid sequence transcripts for clinical samples. All right, so very quickly on clinical evidence. So going back to the DNA part of the study, one of the clinical questions that I've always been interested in as a PhD student in Vancouver in Dr. Richard Harrigan's lab was how DNA was used as, or cannot be, how RNA viruses is used as a protector for biological failure if you detect drug resistance in the RNA. And so in plasma viremia, if there's a drug resistance, you're very likely to respond to a specific drug that matches the drug resistance. And so resistant patterns in plasma predicts treatment success. However, the DNA compartment, as we now know, we didn't know this many years ago, 15, 20 years ago, the DNA compartment, the entire population structure is different. And now with the ability to sequence genome antigen versus defective virus, I went back and asked a clinical question about DNA drug resistance. So using DNA to predict drug resistance has really not been very projective. So there can be drug resistance in the DNA, but the person is not failing. And so people have not really known how and what to do with the viral DNA. We don't actually just have a publication on this. It came in a few months ago or so on DNA genotypic testing and drug resistance.
It's actually still in the press.
So, exactly. So this actually addressed that specific question. So my hypothesis is, and this study is recently funded by Merck, so this grant started in January. So my hypothesis is, well, perhaps DNA drug resistance genotyping is not predicted because all the resistance are either in the intact or the defective compartment. It's not equally archived. And when you're sequencing DNA, you're sequencing just a portion, like protease. And if you only look at protease, and all of the defective viruses that have protease in there would be in your mix as well, and they do not contribute fundamentally there. And indeed, for example, M184 here, all the M184 are archived in the intact virus. We already know in the literature that hypermutations would inflate the count of represented mutations. So we took out the hypermutations, and it's still we didn't take out the hypermutations. We took out genomes that are hypermutated, and it still shows that the intact viruses preferentially archived represented mutations. This is for M184V. We have examples from other drug resistant mutations as well. And this is also fresh out of the lab study, I guess. So we are hoping to dig deeper into this particular analysis, but we currently have 2 individuals who have this pattern that drug resistance is archived in intact, not defective viruses. And if you think about it, this kind of makes a lot of sense. When a person develops drug resistance, the drug resistance is the viruses that replicates. It's reseeds and then becomes the reseeded reservoir. And so those are genome intact viruses. Okay. Meaning that it's more likely, and we know that genome intact viruses are younger, they're more recent than defective viruses because defective viruses are fossils. And so meaning that it makes a lot of sense that resistance mutations potentially are helping the intact compartment versus the defective. But with an asterisk, only if the person failed on a particular regimen and keep finding that particular regimen. And so we're trying to dig into the clinical history of this person to validate that. Right, so that's it. All of this data here, of course, took a village of people, actually global village of people. So a lot of collaborators. I'm very honored to work with groups all over the world. And so thank you very much. And of course, I thank the participants who provide precious samples for all of these studies. Yeah. And the last slide. This is my lab in New York, so we're a very happy group. And we even have a mascot. The second picture there, that's an otter, and this otter's name is Tartar, because the student who owns this otter worked on Tar. All right, so thank you so much for your time.